of training. Did you?
I sent all of them. It's only you can take to work about.
At this minute, we have 5 more minutes or 4. What is the YouTube? Oh, yeah. On YouTube. Subscribe now, this is also just yours. Oh, what?
Please turn.
Good afternoon, everyone, and good morning to some of you. I'm Dr. Sickle, Executive Director of the Joint Clinical Research Center, and I would like to welcome you to this gene therapy awareness webinar series, which are starting today. And this activity is led by the Joint Clinical Research Center, but this is incorporated within the Capacity 2023 project, which is funded by the European and European countries clinical trials partnership, which brings together 5 institutions, including the University of Coimbra, who is the coordinating institution, National Drug Authority, Uganda National Council of Science and Technology, then the JCRC and Muhimbili University of Health and Allied Sciences, MOHAS. So this initiative aims to develop clinical trial oversight frameworks, strengthen regulatory capacity, and enhance the skills of researchers and clinicians in gene therapy. And hence the aim of these gene therapy webinars. As we will hear, gene therapy is the frontier of science. It can be used now to treat or cure several diseases that were incurable in the past. And the first series or the first webinar is being presented by Dr. Selami Emilchi. Please join me to welcome Dr. Selami. Dr. Selami is a staff scientist in the Cellular and Molecular Therapeutics branch at the National Heart, Lung and Blood Institute at NIH. His research focuses on translational gene therapy for hemoglobinopathies, particularly sickle cell disease. And he leverages cutting-edge genome editing technologies, including CRISPR-Cas, and he will again talk about all these things, base editing and prime editing. Dr. Selami has led key preclinical studies in rhesus macaques and humanized mouse models, optimizing delivery methods and evaluating long-term efficacy and safety. He has authored over 50 peer-reviewed publications and book chapters, and is a co-inventor on multiple international patents. His work integrates hematopoietic stem cell biology, gene delivery, and in vivo models to advance curative therapies for genetic blood disorders. Join me again to welcome Dr. Selami, who is initiating the gene therapy webinar series. You're very welcome, Dr. Selami, and over to you. We are listening very attentively. Thank you.
Thank you very much for a kind introduction. Yeah, as you said, I will try to give some introduction to gene therapy, and I'll start with some basics, like some definitions, some basics of the gene therapy, and then I'll try to give some laboratory examples and some clinical data that we see some impressive results that we can now cure some of the diseases that have no cure in the past. But let's start with the root of the problem, which is our DNA. Our body is run on instructions written in our DNA. So these instructions tell our cells how to work. But sometimes there's a tiny, very small mistake, a single change in the DNA that can cause significant problems. And these changes in the DNA can result in some inherited diseases like sickle cell disease, cystic fibrosis, or muscular dystrophy. And these diseases are hard to treat because the problems start deep inside at the genetic level. And today I'll talk about how gene therapy is helping us fix these problems at the source by correcting or changing the mutated genes themselves. So gene therapy represents a new way of treating disease. Instead of just treating the symptoms with conventional drugs, chemotherapies, gene therapy goes to the source of the problem. It aims to fix, replace, or silence the faulty gene itself. And this approach can be life-changing, especially for those people with genetic diseases that have no cure using standard treatments. There are two main types of gene therapy, germline and somatic. Germline gene therapy is done at the embryo stage, meaning the changes are made before a person is even born. The edited gene is passed on to the cells in the body, all of them, including sperm and egg cells. So the changes can be inherited by future generations. But in contrast, somatic gene therapy is done after birth and targets only specific tissues or cells, like blood cells, liver, or muscle. And the changes affect just the person and are not passed on to the children. And all gene therapies currently used in humans are somatic because there are several concerns with the germline editing, including ethical concern, and is not approved for the clinical use yet. So think of a gene therapy like a toolbox. We have different tools for different jobs, depending on the type of genetic problem we are trying to fix. First, we have gene addition. This means adding a healthy copy of a gene to replace or the one that is missing or not working properly. For example, in sickle cell disease, we can add a modified version of beta globin gene that can compensate for the mutated sickle beta globin. Secondly, we can enhance or silence gene expression. This means turning genes up or down without changing the DNA itself. For example, we can silence the Huntington gene in Huntington disease using RNA interference. Or we can use shRNA to silence BCL11A, which reactivates fetal hemoglobin, a very helpful strategy in sickle cell disease treatment. Third, we have gene editing like zinc finger proteins, TALENs, CRISPR-Cas, base, and prime editing. And these tools can make precise change to the DNA itself. A real-world example is, for example, Exocell, which uses CRISPR-Cas to edit an enhancer in the BCL11A enhancer. And again, to reactivate fetal hemoglobin, which alleviates this and alleviates the symptoms of the sickle cell disease. These are very powerful tools, and each one is chosen based on the disease and the cell type and the therapeutic goal. Gene therapy relies on one very critical step, delivering the genetic material into the right cells. So to do so, we use delivery system that we call vectors. And these are broadly divided into two main types, viral and non-viral vectors. Viral vectors like lentiviruses, adeno-associated viruses, AAVs, and retroviruses are the most commonly used in clinical therapy. They are very efficient in entering cells and delivering gene therapy tools. But on the other side, we have non-viral vectors, which include physical methods like electroporation that we are commonly using electroporation at the moment in clinic and a laboratory, or gene guns, and chemical methods like liposomes, nanoparticles, and polymers. Non-viral methods are often safer and cheaper, but can be less efficient. And the choice of vector or the delivery method depends on the several things like the disease and the target tissue and how long the gene needs to be expressed in the target cell and the safety concerns. So long story short, no single delivery method works for every case. The delivery method must be matched to the therapy. But let's take a step back and look at the bigger picture of the gene therapy research over time. In 1972, gene therapy was first proposed as a treatment idea, and it took almost 2 decades before the first human trial began in 1990. And in 1999, the field faced a very significant problem. A patient death during a clinical trial raised serious concerns about the safety. And this led to stricter regulations and a pause for reflection. Progress resumed not until early 2000s. And by 2017, the first gene therapy was approved in the United States for a genetic eye disorder. In 2019, gene therapy was approved for spinal muscular atrophy in very young children which was a major milestone. And today, we have over 2,500 gene therapy projects in development worldwide, and the approvals by the FDA continue to grow. So we have seen how gene therapy has progressed over the last 50 years from an experimental idea to a reality with approved treatments. But how do we actually deliver these therapies in real work? One of the most successful strategies so far has involved targeting a special type of cells in the body, bone marrow, hematopoietic stem cells, or in short, HSCs. These cells are at the root of entire blood and immune system. And if we can fix these mutations in HSCs, we can fix the many diseases at its source, such as storage disorders, immunodeficiencies, and hemoglobinopathies, including alpha beta thalassemia or sickle cell disease. As I'll be giving a lot of gene therapy examples on sickle cell disease, I would like to give a very brief information about the disease. It's one of the best known single gene disorder, and also one of the first where gene therapy has moved from a clinical trial to real practice with approved treatments. Sickle cell disease was first described more than 100 years ago by Dr. James Herrick, who noticed these oddly shaped red blood cells. Later in 1949, Linus Pauling described this as a first true molecular disease caused by a single point mutation in the beta globin gene. And the mutation leads to abnormal hemoglobin that forms rigid polymers when oxygen concentration is low, causing red blood cells to sickle, block blood flow, and cause severe complications, including major organ damage and significant morbidity and mortality. Although it's considered rare in the United States, affecting almost 100,000 people, it affects millions globally, especially in sub-Saharan Africa, India, and the Middle East. For many years, our treatment tools were limited. We've used medications like hydroxyurea, which can reduce symptoms by increasing fetal hemoglobin, but it doesn't address the root cause of the disease. In some cases, we've turned to bone marrow transplant from matched donors, and that can actually offer a cure, but it is very risky in terms of graft rejection, graft versus host disease, and mortality, and not available to most patients. Like more than 80% of the patients cannot find a suitable match sibling donor in the United States because suitable donors are hard to find. But now with the gene therapy, we are entering a new era. Instead of relying on donor cells, we can use the patient's own stem cell, own hematopoietic stem cells. In what's called ex vivo gene therapy, we take those cells out of the body, fix the mutation in the lab, either by adding a new gene or directly editing the faulty one, and then return the corrected cells to the conditioned patient. And this is how both FDA-approved treatments like lentiglobin and exa-cel work. So looking ahead, researchers are now exploring in vivo gene therapy, where the treatment is delivered directly into the body, potentially making the process even simpler. The bottom line is we're moving beyond managing diseases like sickle cell disease to actually curing it. And for many patients, that could mean a one-time treatment with lifelong impact. One of the most important questions in gene therapy for sickle cell disease, for example, how much correction do we actually need to see a clinical benefit? Based on NIH allogenic transplant data and mathematical modeling, it turns out that the answer might be surprisingly modest. The study in our lab by Fitzhugh and colleagues showed that just 20% donor myeloid chimerism could lead to a much higher proportion of the healthy red blood cell, because the corrected cells survive longer than sickle ones. So the real takeaway here is this: we don't need to correct every stem cells. Even a relatively low level of gene correction may be enough to provide meaningful clinical benefit. And that raises an exciting question. Can we reach that 20% threshold using gene addition strategies with using the patient's own hematopoietic stem cells? So speaking of gene addition is one of the foundational techniques in gene therapy. Instead of fixing the mutation itself, we simply add a new working copy of the gene to compensate the mutated one. And this new gene is delivered using a delivery vector, often a virus, generally lentivirus these days, and taken up by the patient cells. Gene addition works well when the disease is caused by a loss of function, meaning the faulty gene that just doesn't make enough of what the body needs or make a unuseful one. And it's not ideal when the mutation causes a toxic gain of function. But in sickle cell disease, for example, gene addition has shown great promise, especially in clinical trials using lentivirus. So we use integrating viral vectors like lentiviruses as delivery vehicles. These vectors are engineered to carry the healthy gene, and they act like a Trojan horse, entering the patient's hematopoietic stem cells. Inside, they deliver the therapeutic beta globin gene, for example, which becomes part of the cell's own DNA. Because it's integrated, it gets passed on as the stem cells divide and produce new blood cells. This is really important for conditions like sickle cell disease because we need the new gene to be active in red blood cells, and we need that expression to be sustained and high enough to make a clinical difference. In early 2000, gene therapy showed success in treating X-linked SCID, a fatal immunodeficiency. Using gamma retroviral vectors, patients who would have died in infancy developed functional immune system, and it was a major milestone. Unfortunately, some patients later developed leukemia. The retroviral vector had integrated near the proto-oncogene, namely LMO2, turning on some genes that it wasn't supposed to be expressed. And this serious side effect called insertional mutagenesis was a significant problem to overcome at the time. After the safety setbacks we saw with the early retroviral vectors, especially the cases of insertional mutagenesis in SCID trials, there was an urgent need to develop safer tools. That's where lentiviral vectors come in. These vectors are derived from HIV, but they are heavily modified to be non-replicating and safe. On top of that, we can now use tissue-specific promoters and enhancers, so the gene is only turned on in the right cell types. And these advances dramatically reduced the risk of insertional mutagenesis and made it possible to move forward confidently with lentiviral gene therapy in diseases like sickle cell disease. Once the safer lentiviral vectors were developed, scientists began using them to treat diseases like beta thalassemia and sickle cell disease. And this slide shows early results in mouse models when researchers delivered that therapeutic beta globin gene using lentiviral vectors, and they saw high level of healthy hemoglobin and reversal of the disease symptoms. And these studies proved that the strategy worked and opened the door to clinical trials in human. And this led to the HBG 206 trial, which was partially conducted in our laboratory. And this trial tested a lentiviral gene therapy product called lentiglobin. Patients with severe sickle cell disease had their own hematopoietic stem cell collected, and these cells were then genetically modified outside the body using a lentiviral vector that delivered a modified version of beta globin gene with higher anti-sickling properties, namely T87Q. After conditioning with busulfan to make a space in the bone marrow, the corrected stem cells were then reinfused to the patient. The goal was for these cells to engraft and begin producing red blood cells that contain therapeutic and anti-sickling hemoglobin. And the results were very promising, especially in Group C, where patients received a refined manufacturing process and mobilized stem cells. As shown in this chart, patients began producing high level of T87Q, the gene therapy-derived anti-sickling hemoglobin. And in many cases, over 40-50% of their total hemoglobin came from the therapeutic gene, which is considered more than enough to prevent sickling and reverse disease symptoms. These findings helped support the FDA's approval of this therapy, marking a milestone in the treatment of sickle cell disease. Patients in Group C produced high level of therapeutic gene, yes, but the real question is, did it make a clinical difference? The short answer is yes. This chart shows the number of severe vaso-occlusive events before and after lentiglobin therapy. On the left in blue, you can see the frequent crisis before treatment. On the right in yellow, the same patients after receiving the gene therapy had almost no events. The median number dropped from 3.5 per year to 0. For many of these patients, it meant a complete end to painful, life-disrupting episodes. Sometimes, instead of fixing or replacing a gene, we can simply adjust how active it is, kind of like turning the volume up or down. One of the approaches is enhancing gene expression. That means boosting the activity of helpful genes. For example, in sickle cell disease, people stop making a type of hemoglobin that protects against symptoms, but scientists found a way to turn that protective gene back on by blocking another gene that was shutting it off. In addition to these, we have also the option to silence harmful genes. Some diseases are caused by a gene making a toxic protein. Here we can use tools like RNA interference or CRISPR to reduce or stop that gene from being read. So gene therapy can help not just by fixing broken genes, but by rebalancing gene activity to help the body function better. Here's a really powerful example of gene expression modulation used to treat sickle cell disease. In sickle cell disease, the red blood cells become rigid and sticky because of the mutation in adult hemoglobin gene. But before birth, our bodies produce a different form called fetal hemoglobin, or HbF, which doesn't cause sickling. After birth, natural genetic switch turns off the fetal hemoglobin and activates adult hemoglobin instead. Scientists discovered that a gene called BCL11A is responsible for the switch. It acts like a brake that shuts down fetal hemoglobin production. If we block or silence BCL11A, we can remove that brake and the body starts producing fetal hemoglobin again, even in adults. Clinical studies have, so why fetal hemoglobin is important? Because the clinical studies have shown that people with naturally higher fetal hemoglobin levels tend to have fewer complications and live longer with sickle cell disease. By using gene therapy to target BCL11A, we are not fixing the mutation, sickle mutation directly. We are reactivating a natural protective mechanism that can reduce the symptoms and improve survival. In this regard, patients with sickle cell disease had their blood stem cells collected and modified using a lentiviral vector, carrying a short hairpin RNA, or shRNA, if you will, specifically designed to silence BCL11A. After receiving their modified stem cells back, patients began producing increased level of fetal hemoglobin. For most, fetal hemoglobin levels rose above 20%, and some maintained even higher at the long-term follow-up. More importantly, these patients saw a sharp reduction or complete elimination of disease symptoms. Many had no severe pain crisis or transfusion requirements after treatment. This trial showed that post-transcriptional silencing of BCL11A is not only biologically effective, it is also clinically meaningful. So far, we've looked at the ways to add genes or turn existing ones on or off, but what if we have to go straight to the DNA and we need to edit the mutation itself? That's where genome editing comes in. Over the years, tools like meganucleases, zinc finger proteins, and talons have been developed to cut DNA at the precise spots. But today, the most widely used tool is CRISPR-Cas, which uses a simple guide RNA to direct a molecular scissor to the target. It is fast, accurate, and easy to program, making it the foundation of modern gene editing therapies. All of these tools function by introducing a double-strand break, which the cells then repairs, enabling us to insert, delete, or correct specific positions at the DNA. CRISPR stands out because it's very simple, flexible, and cost-effective with high editing efficiency and the ability to target multiple sites at once. Earlier tools like TALENs and zinc finger proteins still work well, but are more complex and harder to scale up. The accessibility of CRISPR has truly democratized genome editing. It is now used everywhere, from high-level laboratories, high-tech laboratories, to online shopping. Here's how CRISPR actually works inside a cell. The guide RNA leads the Cas9 enzyme to a specific DNA sequence where it makes a double-strand cut. And after the cut, the cells can repair the break in two ways. One way, namely non-homologous end joining, disrupt the gene by adding or deleting random sequences. And the other allows for precise correction using when a donor template or a repair template is used. And this is namely homology directed repair. This system let scientists either turn off a gene or fix a mutation, making it incredibly versatile for treating genetic disease. I'll give an important example where in sickle cell disease, for example, CRISPR is used not to fix the sickle mutation directly, but to disable a regulatory element in the BCL11A gene. And I mentioned this a couple of slides ago. The BCL11A is controlling the fetal hemoglobin expression, which is a very important protective protein for sickle cell disease, because fetal hemoglobin is not sickling and preventing red blood cells to sickle in sickle cell disease patients. And using this knockout system, silence BCL11A in red blood cell, which releases the brake on the fetal hemoglobin, helping prevent sickling. Editing just this enhancer is enough to restore the protective fetal hemoglobin levels. And this kind of study is led to an FDA approval for CRISPR use for sickle cell disease patient in December 2023. An approved therapy is called exa-cells. That patients with severe sickle cell disease had their own stem cells edited to knock out the BCL11A enhancer. And after receiving their edited cells, most patients experienced complete resolution of the severe pain crisis. And this was a landmark moment, not just for sickle cell care, but for the entire field of genome editing. It was the first time CRISPR has gone from the lab to the real patients with life-changing results. So I'll give another example other than the sickle cell disease. So in the HIV infection, the virus typically enters host cells through the CD4 receptor and a co-receptor called CCR5. But there are some individuals naturally carrying a mutation in this CCR5 location, which makes them resistant to HIV infection. Even though they were exposed to HIV viruses, they are not yet infected because they don't have this core receptor functionally active for the HIV infection. Historically, allogeneic stem cell transplant from CCR5 mutant donors are being used and have led to a long-term remission of HIV in many patients. But this approach is high risk and only suitable for those with suitable donors. A safer and a more scalable alternative is gene therapy. We can now isolate patients' own T cells or stem cells, and we can knock out the CCR5 gene using tools like CRISPR or zinc fingers and then reinfuse them. And these edited cells generate immune cells that are resistant to HIV infection. And this concept was first clinically tested in 2014 using the zinc finger nucleases to disrupt the CCR5 and CD4 T cells. But more recently, CRISPR-edited HSCs were transplanted into a patient with HIV and acute lymphocytic ALL in China. And while full viral eradication was not achieved, the study showed the feasibility and safety. Ultimately, this strategy reflects how gene editing is not only being used to fix inherited disorders, but also create resistance to infectious disease. CRISPR-based editing has delivered impressive clinical results, as we've seen in the last couple of slides, especially in sickle cell disease. But like any other powerful tool, it comes with some risks, mainly due to the way it works. By cutting the DNA from both strands, double-strand breaks caused by CRISPR-Cas9 can result in unintended consequences, large deletions, chromosomal rearrangements, and even chromatosis, where chromosomes shatter and reassemble incorrectly. These risks are especially relevant in stem cells. As shown on the right, we found that editing cells can especially reduce editing by the CRISPR-Cas and causing a double-strand break can especially reduce the stem cell quality or survival. To overcome this, newer tools aim to edit the genome editing without breaking it. So how do we fix mutation without breaking the DNA? Base editing, a more precise, safer approach that doesn't rely on creating double-strand breaks. Base editing is like pencil and eraser for DNA. It uses a modified version of CRISPR system that can directly convert one letter of DNA into another. For example, changing a G to an A without making a double-strand break. This is done using enzymes called deaminases, which chemically modify DNA bases. And the result is highly targeted in the editing window with a much lower risk of the complications we saw with the traditional CRISPR system. This precise approach is especially promising for diseases like sickle cell disease because there is only one mutation. So if we can convert that mutation in sickle cell disease, then we can convert the disease. And after converting the mutation with the base editing, we can restore normal hemoglobin and reduce sickling. So in the images here, you can see that the base edited cells behave more like healthy red cells and produce much less sickled hemoglobin. Editing was also efficient across different blood cell types, including stem and progenitor cells. And this shows that the base editing isn't just safe or safer, let's say, it is also effective and could play a key role in the next generation of gene therapies for sickle cell disease. So base editing gave us a way to rewrite single DNA letters without breaking the strand, yes, but what if we want to make more complex changes, like inserting or replacing a short sequence? That's where prime editing comes in. Think of it as a search and replace function for DNA. Prime editing is one of the newest and most precise genome editing technologies we have. It uses a modified Cas9 nickase, so it nicks just one strand of DNA instead of cutting both, and it is fused to reverse. It is fused to a reverse transcriptase enzyme that can write a new genetic code directly onto the genome. A special guide RNA called prime editing guide RNA or PEG RNA tells the editor where to go and what changes to make. And the results, we can now insert, delete, or swap short DNA sequences with great accuracy without causing double-strand breaks. In a study by David Liu's lab, where prime editing and base editing has been developed, researchers used prime editing to correct sickle cell mutation in mouse models. You can see the original sickle mutation in the red text and how it was precisely changed to a healthy version using a prime editor. After editing, the mice produced more normal beta globin and showed dramatic reduction in sickle red cell. And these results suggest that the prime editing may one day allow us to repair sickle mutation itself without cutting the DNA. While gene editing tools like CRISPR, base editors, and prime editors are transforming our ability to fix disease-causing mutations, their clinical success still relies on one critical step: getting the edited cells to engraft. And that brings us to an equally important part of the puzzle, how we prepare the patient's body to receive these corrected cells. To successfully replace disease cells with edited ones, we need to first clear space in the bone marrow and open a niche for the newly edited cells. This is done using high-dose chemotherapy like busulfan, which creates an open niche room for the modified stem cells to engraft and rebuild a healthy blood system. This conditioning step is historically done with the chemotherapy like busulfan and previously done by the total body irradiation, but it is very active and effective in conditioning, but it is not selective to hematopoietic stem cells, and it's very toxic. It can damage fertility, injure organs, and increase the secondary malignancy risk. And these serious side effects could prevent many patients from benefiting from the gene therapies, especially children or those with existing complications. In this regard, our laboratory works on alternative conditioning regimens, and one of these approaches is to use antibody-drug conjugates that targets only hematopoietic stem cells or just the blood cells. One of those approaches is using the target protein expressed in blood stem cell that is conjugated with a toxin, namely CD117. And CD117 is one of those proteins that are expressed on hematopoietic stem cells. And we've previously showed that this CD117 ADC method, coupled with a gene therapy, not only achieves good stem cell engraftment and induces significant fetal hemoglobin that is very protective for sickle cell disease, but also preserve fertility in animal models. In fact, most animals conditioned with CD117 ADC were able to reproduce, unlike those treated with busulfan. Another promising approach is CD45 ADC, which targets all immune and blood-forming cells, including hematopoietic stem cells. This broader depletion can help make space for new cells and reduce the risk of immune rejection. Immune rejection, of course, is not a case for autologous stem cell transplantation, but if we want to increase the gene therapy methods to the allogenic setting, then maybe we might need to use CD45 ADC rather than CD117 ADC, which is potentially expanding the gene therapy access to more patients. We developed a strategy where we multiplex edits BCL11A enhancer and showed that its fetal hemoglobin-inducing activity is superior to any known strategy, including the FDA-approved approach. We then used this dual targeting edited BCL11A editing strategy combined with CD45 ADC for conditioning in a non-human primate study. And this showed a strong reactivation of fetal hemoglobin while maintaining safety and engraftment. Importantly, when we compare CD45 ADC to busulfan conditioning, we saw similar levels of gene editing and therapeutic fetal hemoglobin levels in animals, suggesting we can achieve the same clinical benefit with much less toxicity. While antibody-drug conjugate-based strategies like CD117 ADC or CD45 ADC offer major improvements over traditional genotoxic conditioning like busulfan, they still require elimination of both edited and non-edited hematopoietic stem cells. But what if we could selectively spare the edited cells while clearing the rest, like unedited cells, without needing a toxin payload? So we decided to use a naked antibody and use a multiplex base editing strategy to be able to spare the edited cells from CD117 conditioning. That's where the concept of escape study comes in, a base editing strategy designed to make engineered stem cell invisible to the depleting antibody. Our team used base editing to change the epitope on CD117, the receptor targeted by the depletion antibody, and in unedited stem cells, anti-CD117 blocks CD117 signaling and the cells die. But in the edited cells, the modified epitope prevents the antibody from binding, so the cells escape depletion, maintain normal signaling, and survive. Importantly, these surviving stem cells also carry therapeutic edits that upregulate fetal hemoglobin, creating a dual benefit. The next question is, can we actually select edited cells after transplantation? So this is very important. After making a transplantation, you have only one shot. If the editing, the threshold that you were aiming to reach is not good enough, then what you're going to do? So you have to clear everything and you have to find a different solution. But what if we have a solution to selective, positively select the edited cells in the body after transplantation? So this multiplex base editing can provide this. In a non-human primate model, we see a rapid and complete turnover of the unedited erythroid cells after transplantation. Fetal hemoglobin producing F cells increased sharply, reaching about 60%, and the gamma globin levels reached to 40% by the same time point, around 8 weeks post-transplantation. And this early and robust reactivation of fetal hemoglobin demonstrates the potential for a fast-acting clinical benefits in patients. We've seen so far that the gene therapy can result in remarkable clinical outcomes, but this comes at a price, literally. Lentiviral gene addition and CRISPR-based therapies are currently priced around 2 to 3 million per patient. An allogeneic transplant, still costly, ranges from $200,000 to $500,000. And these price tags raise a critical question about how we define value in healthcare and what's feasible for patients and healthcare systems. And yet, the burden of sickle cell disease is greatest in low and middle income countries, particularly sub-Saharan Africa and India, where the vast majority of the affected births occurs. As the global population grows, so will the number of newborns with sickle cell disease. This underscores the need for affordable, scalable therapies that can reach beyond high resource settings. Let's say if you want to assign all the transplantation unit in the United States, which is a developed country, for just sickle cell disease patients, we can only treat 10,000 patients per year, meaning that to finish to cure all the patients in the United States, we need 10 years to use all the transplantation units in the United States to cure all the patients in the United States. And in the United States, we have only 100,000 sickle cell disease patients diagnosed, but most of the patients are not in the United States. They are in low and middle income countries. So this shows that these techniques, gene therapies that we have now by the FDA approved, is very effective, but it's not scalable at the moment. So we have to find some methods that we can scale this up. And what do we do with the current ex vivo gene therapies? We collect the stem cells from the patient, modify them outside the body, and then reinfuse them after conditioning. While it's very effective, the approach is intensive, hospital-based, and resource-heavy, contributing to the high cost and the limited access. By contrast, in vivo approaches where gene delivery happens directly inside the patients could transform accessibility. Instead of collecting and edited cells outside of the body, we can use targeted vectors to deliver genetic payloads directly to the stem cells. This strategy could eliminate the need for hospitalization and ex vivo stem cell manipulation altogether. And this is not just theoretical. In vivo genome editing has already entered the clinic, from treating transthyretin amyloidosis and hereditary blindness and rare liver disorders and many, many others. And these pioneering studies show us that delivering genetic therapies directly into the body is not only possible, but it is already changing many lives. So to wrap this up, Gene therapy has evolved from a scientific idea to clinical reality. Tools like CRISPR, base editors, prime editors, and silencing strategies are enabling us to precisely correct and regulate disease-causing genes. The next frontier is and should be in vivo gene therapy, which offers hope for simpler, more equitable solutions. But to truly fulfill the promise of genetic medicine, we must also address cost, infrastructure, and global access. So with that, I would like to thank to my laboratory and thank you for listening. And I am happy to take any questions you may have.
Thank you very much, Doctor. We will start with some questions which people have already sent us by email, but there are also some questions online, and then we'll allow people to put up their hands if they have additional questions. Some of the questions which were asked, I think you've answered. People were asking, how do you define gene therapy? I think you have done that. There was a question after the webinar series, is there any kind of documentation or certificate that will be awarded to members who attended the series? So we have a series of about 10 webinars and attendance of 75% of these webinars. Yes, we will provide a certificate. There was a question, are there any other platforms for gaining more knowledge about this subject in regards to healthcare? Because I see many people registered, but we only have about 270 online. So after the webinars, we will create a page on the capacity website. Again, we will be circulating this, and also on the JCRC website, so that people who are not able to attend can go back and listen to this very innovative work that is being done, and also to understand the basics. The other question was, Sickle has been highlighted among those where gene therapy is promising. Are there any other illnesses that would possibly be treated or cured using gene therapy? Over to you, Dr. Selami.
Yeah, so gene therapy is currently being used for many different kind of diseases, including like muscular dystrophy or some other genetic diseases. So I only highlighted the sickle cell disease or hematopoietic stem cells because that's my expertise. That's where I'm working on. But it's been used for many other diseases, including several cancers. You know, for example, you can, there is a very important area that we can actually use gene therapy to create CAR T cells that can target many different kinds of cancers. And at the NIH and many different places in the world, this CAR T therapy is being used successfully, eliminating the several different kind of cancers. And the results are now showing significant promise, and they are being transferred to clinics as well. So I highlighted the use for the inherited diseases like sickle cell disease, but as you might remember, I also showed that it can be used for the infectious disease as well. If you prevent the infection mechanisms like the CCR5 transduction of the HIV, then you can prevent HIV transmission, or you can provide an HIV resistance. So yeah, it can be used for many different kind of disease, not just inherited disease, but infectious disease and cancer and many others.
Lami, I think very importantly, we are also looking at using gene therapy for cure for HIV. We know we don't have a cure, we have treatment, but funding for this treatment is very unreliable. So certainly HIV is one of the infectious diseases that we are looking at. So there was someone who was asking to share the PowerPoint. We will not share the PowerPoint, but there is also live streaming, so there will be a YouTube so that you can listen again, and as you see the slides, there will also be explanations that Dr. Selami has given. And these links, again, will be on the website page of Capacity, but also on JCRC. There is a question about what methods are used to ensure precise, safe, and durable gene editing without off-target effects or immune complications. Do we have a library catalog of side effects from short to long term in animal and human studies? That's from George.
Yeah, that's a very important question. So we are trying to do some changes in the DNA, but changing DNA is risky, right? As I showed, using CRISPR-Cas, we show that we can provide some significant improvements in sickle cell disease patients, but using CRISPR-Cas9 provides several unintended consequences that can result in some other problems like secondary malignancies. So that's why the genome editing technology is being improved every day, day by day. Like when we had it first in 2013, we didn't know the side effects and off targets. And but we started to understand that there was some significant problems by standard CRISPR-Cas9. So that's why we developed base editing. And after developing base editing, we saw that, okay, base editing is good and provides limited, off-target activity, but it's not enough for all kind of editing systems. So we developed another one, the prime editing, and still we're trying to improve it more. But to understand what are the effects, we have to use some preclinical models like non-human primate models or humanized mouse model to understand the potential risk of a therapeutic approach before putting into the clinical trials. But even with those studies, preclinical studies, it's not possible to understand every possible side effect. So that's why we still need clinical trials too. But hopefully, eventually at the end of clinical trials, we will understand the full safety and efficacy of the approaches, and we will use this in our clinical systems. And there was this question, is there any catalog that we can see all side of effect? No, and there's not, but also off targets and the problems is case by case. So it depends on the guide RNA that you are using. It depends on the target that you are aiming in the DNA. So it changes case by case.
Thank you very much. And also, I think for the long term, patients in gene therapy trials haven't been followed up that long. But now FDA mandates that patients in these gene therapy trials are followed up for at least 15 years. So that while we are looking at short term, we can also look at long term side effects.
Exactly.
So the other question is, why not use base editing to treat HIV?
We use, the whole idea is to disrupt CCR5 sequence. And disrupting CCR5 sequence can be done by using standard CRISPR-Cas9. And if you create a double strand break, then you will disrupt the sequence, and that's good. And that's how we do in the clinic now. But newer ways, for example, if we change the sequence by base editing, then it's still fine, and it seems to be a better option. And now it's being tested in the non-human primate models. And for those who are familiar with the literature, for example, you can go and check the Hans-Peter Kim's laboratory results, and they use the base editing for HIV treatment.
Yeah, thanks, Selami, because we have had at least now 9 patients who have been cured of HIV by bone marrow transplant using donors who had Delta 32 deletion. And so the most exciting or the most feasible way to replicate that is through gene therapy. So I don't know why we are not yet there with this Delta 32 deletion, either with base editing or prime editing or whatever.
Yeah. I hope improvements are going to be faster in the upcoming days. You know, one reason that is a very significant setback is the cost. As I showed, it's very expensive. Not a lot of institutions can cover or find money to do this kind of research. So we have to find a way to lower the cost to make it more accessible for the whole world.
Yeah, but the research, there are many companies doing gene therapy now, and
Sure.
I thought this, and they are getting money either from investors, but this replication of the 9 patients who have been cured by BMT, I think shows a more direct path to HIV cure modality.
Sure, I agree.
Okay, there is a question from Enoch. Is there any way we can get access to the content presented during the webinars for further revision and contextual analysis? Yes, there is, we are doing live streaming, so there is a YouTube, which people can go back to. It will have the slides and the presentation as given by Selami. And we will also be putting this on the Catalyst website, but also the JCRC website, so that people can link in and see and review. Because this was very interesting, maybe it was a bit fast, and yeah, revision certainly to understand what is happening and to understand the basics is very important. I think there is a hand. Maybe we take one question, then we take the hand. I would like to request for the recordings. Again, I've already, I think, said that. Before Francis comes in, he's here in the room, he'll ask his question. Let us ask the person who is putting up their hand. Charles Odongo.
Hello, can you hear me?
Yes.
Very clearly.
Yeah, thank you for such a wonderful presentation. Clearly very enlightening. I imagine that CRISPR-Cas9 is a highly specific procedure, but how exactly does it lead to other adverse effects like cancer?
So far, there is no report showing that, you know, secondary malignancies like cancer is the result of the CRISPR off-target activity. So off-target is like a concept, and because it's also a change, an unintended change in the DNA, so it scares people. And scientists are very protective and precautious about this kind of issues. So we are trying to find ways to have less off-target activity, more on-target activity, even though we don't have any preclinical or clinical data showing that CRISPR is directly causing any cancer. But of course, because there's a DNA change, chromosomal rearrangements, and changes in the chromatin can cause theoretically significant problems. That's why we have to be very careful in designing new therapies, especially if we are changing DNA at the unintended sites. But there is no any preclinical or clinical studies showing that the CRISPR can cause, cancer at the moment.
Thank you.
Okay, thank you, Charles. John Bosco, you also have your hand up.
Yes, good evening. I had put the question in the chat box, but I think the moderator had through an oversight not seen it. My issue is on any ethical concerns as far as gene therapy is concerned, and how do we intend to mitigate that? Thank you.
I, I couldn't hear you exactly. Can you repeat it, please?
Do we have any ethical concerns as far as gene therapy is concerned? And if so, how do we intend to mitigate that?
Yes. So the ethical concerns are mainly remaining on the germline editing. So in preclinical studies, it's been shown that it's possible, but the ethical concern still remains for the germline editing. So it's not approved for human use, even in clinical trials. And so these ethical concerns are not easy to overcome because, because it's also relies on the social characteristics and the ethical standards. So I don't think most of the countries are trying to handle this. But in the somatic editing system, there is not any ethical concerns at the moment, other than trying to be socially fair, because this kind of therapies should be available for all, not only for a small portion of the society, and try to make it this for all. We need to make it more expanded and available for clinical use. And how this is going to happen is the more we do, the more it's going to be available for clinical use. And the more we work on it, we are going to find cheaper ways to produce more and make it cheaper and applicable for everybody in the world.
Thank you.
Is there any other question? Maybe there's one in the chat saying that is fetal hemoglobin anti-inflammatory? Recently saw a young man, 21 years old, a military considered referred to the evaluation of severe anemia with no associated pain. We had received a transfusion by the primary doctor. Surprisingly, we had a sickle cell seen on his peripheral blood film, a high serum LDH and reticulocyte. The hemoglobin electrophoresis revealed for 53% sickle and 25% fetal hemoglobin. He was very surprised at this sickle cell disease diagnosis since he had no past history of painful episode. It was the severity of the military exercise that preceded the disease symptoms, and he had not been on any prior therapies. Yeah, that's how we understood fetal hemoglobin is important, because there are some patients, they didn't even know that they are sickle cell disease patients on a routine check. So they were discovered that sickle cell disease, just like this patient, on a routine check, because they have very high level of fetal hemoglobin, like these patients, over 20%, 30%, that prevents them to have pain crisis, vaso-occlusive crisis, because this is protective. So the whole idea of gene therapy in this regard is try to find a ways to increase fetal hemoglobin levels so that it can be protective. Can I pass on the genes from gene therapy to the next generation? For example, if I have sickle cell, can I produce a child with A after gene therapy? No. Gene therapy is done on the somatic cells, meaning that it is only for you, because we are not doing the gene editing on the germ cells, on sperm. The sperms and the eggs still carry the sickle mutation. So even though you have gene therapy and you're cured from it, if you have children, and there's a possibility that children can have sickle cell disease. Are there any observed racial, age, gender difference in response to gene therapy? So in terms of age, the younger generations for sickle cell disease patients, for example, younger age are better, but not in terms of gene therapy, in terms of conditioning, because the older you get, the longer you have sickle cell exposure, meaning that you have more damaged organs, which makes it less resistant to the conditioning or chemotherapy. But in terms of racial or gender, that's not something that I know of. Any comment on the behavior of the edited cells, especially in regards to the regulation of cell division, as it could lead to cancerous cells? This is not proven yet by the preclinical or clinical trials. They seems to be fine as long as it passes the preclinical research. It seems to be doing fine, but as Dr. Sisi said, we need to have very long-term follow-up, and we are just in the beginning of this new era, so we have to wait and see all possible side effects in the long term. How will the high cost barrier to be overcome? So this is one way of the science. The science always start with from a higher price because it's not produced in bulk, but the more we are interested and the more we work, always it gets cheaper, but it's a long term, it's a marathon. So there are a lot of laboratories trying to make it in a bulk production. And one of the problem with the high cost is having an infrastructure that is capable of doing transplantation in a GMP, good manufacturing process environment. And this is very expensive. If we can find ways to do in vivo editing instead of outside the body, then we can make it cheaper. Consider it like a gene therapy, like a COVID shot. If we have only one single infusion that is taking care of the editing inside our body, then it can be cheaper. And there are many research showing that this is possible.
Adrian has his hand up. He actually has two hands up, Adrian. Adrian? Adrian, do you want to ask your question?
Maybe I can answer one of the questions on chat. Overall, when you compare non-viral vectors to viral vectors, which one of two offers a safer route with better outcomes? So it's also case by case. There are some immune response to viral vectors. So generally, it is considered that the non-viral vectors are safer, but they can be less effective. But nowadays, we are using lipid nanoparticles or polymers that are very effective, and I'm expecting them to be in the clinical trials soon.
So there is another question, Dr. Salami, from Alex. Like sickle cell disease, does one remain infective, and are there a possibility of passing on HIV after gene therapy?
Is the question that after gene therapy, is it possible to have HIV infection? Is that the question?
It's a bit confusing, but Alex had his hand up. Alex, can you ask directly?
Okay, thank you, Doctor. My question was on the fact that you said there still remains a possibility of passing on sickle cell disease even after the treatment with gene therapy. So my concern was, is it the same case with HIV, that when one is actually treated and cured with gene therapy, can still pass on the HIV virus? Thank you.
You're asking that whether after the gene therapy you can still transmit HIV to some other person? Is this the
Yes.
question?
Yes, after being cured.
Yes. So
Yeah.
yeah, maybe Dr. Sisi can answer that.
Yeah, because we do not have yet a gene therapy to cure HIV. But we are working very hard. The whole world is working very hard to get a cure. However, what cure means with gene therapy, if we have it, is that people who are cured are able not to have any viral load measured in their blood when they get off antiretroviral therapy, okay? So transmission of HIV is based on a viral load, and you have also read, or you will read studies where they say if viral load is below 1,000, the possibility of transmitting HIV to another person is very limited. It's not zero completely. So if we have a gene therapy that can cure HIV, that means people will have no viral load in their blood. Therefore, they are not able to transmit HIV. I hope that is clear. Adrian, you were not able to talk. Can you unmute Adrian?
Are you able to hear me now?
Yes.
Okay, wonderful. Thank you so much for the talk. It was very enlightening. My question is actually with regards to the humanized mouse models that are being used for a lot of these gene therapies. Is there a consensus, for example, for something like sickle cell disease? Are you using a consensus sequence? Because obviously there's many haplotypes with regards to sickle cell disease. Plus, I mean, there's epigenetics and a lot of pleiotropic effects as well. So how do we know that, let's say, the mouse model that we're using is necessarily the best representative of a consensus patient, right? I mean,
Yes.
obviously it's never perfect, but how do we know that?
Yes. The short answer, we don't. Even in clinical trials, once we have therapies, that doesn't reflect the responses for all patients. So every patient is case by case. But what are we trying to do with the animal models? We are trying to increase the possibility of a positive outcome. So how it goes is we first test something in our cell lines. If we give an example from a sickle cell disease perspective, we use the red blood cells or differentiate stem cells into red blood cells and test the gene therapy or the genome editing in those cells and to see whether the outcome is positive or not. If the answer is yes, then we go next to the mice model. And we have different mice model like Towns mice that have a sickle sequence. And we have also some immunocompromised mice that we can use human cells. And we test our strategy in mice models, which takes around 6 to 12 months. And then if we find a positive result, then we go to the next section, which is non-human primate models. And in those non-human primate model, we don't have a sickle non-human primate model, but we're doing a transplantation model. And we're trying to understand whether our system is working in a larger animal model because it has a longer follow-up. For example, I have some animals transplanted with CRISPR-Cas that have longer than 10 years follow-up. And after this, we design a clinical trial, and the clinical trials goes from phase 1 that you're investigating the efficacy and safety in phase 2, and then the scalability with the phase 3. And even at that point, there is no way to make 100% sure that this is safe and it's going to be perfectly working for the next patients. So all these processes are to increase the possibility of the positive outcome. We can't just find an idea and go and do in the clinic. So this is the way that how a new approach is being approved and used in the clinic. But in terms of the mice models, they are valuable. They are not reflecting the human physiology at all, but they are reflecting the cell behavior inside an in vivo environment. So it's better than the cell line models, but it's not any better than, or not reflecting the human physiology at all.
Okay, thank you.
Yeah, thank you.
Can you hear me, Dr. Sebani?
Yes.
Yeah, there is a question in the chat. If I undergo the gene therapy procedure, do I inherit the traits from the gene?
We don't inherit if it is done on the hematopoietic stem cells or somatic cells. As long as it is not done in the embryo, then we are not inheriting any gene therapy to the next generation.
Okay, there is someone called Sickle Cell Program. They have their hand up. Can you ask your question? Sickle Cell Program, wherever you are.
Thank you, Professor Sisi. I hope you can hear me. This is Daima.
Yes, we can hear you.
Thank you. So I have a question. You mentioned about the next phase being large production of seeing ways that we could sort of like lower the price by having large production. So I think my question is, with having no clinical trials currently in Africa for gene therapy, either on HIV or sickle cell disease, so does it mean the next phase wouldn't be doing clinical trial, of course, with the advancement that have already been done elsewhere in the world? So does it mean once those trials have already been done either in the US or in the UK, does it mean the next phase for us is just clinical practices? So we won't have a chance of implementing clinical trials related to gene therapy, either sickle cell disease or HIV. So I'm a little bit confused in terms of what does it mean for us? Are we going to have an opportunity or access to do clinical trials with the patients that are here? Or does it mean we are going straight to cure? Thank you.
Yeah, I think, clinical trials are very important. That's why they're, even though we have like a phase 1, phase 2 clinical trials approved, we still need the phase 3 or phase 4, even like when it is in the clinic, right? So because multi-center clinical trials are very important because as there was this question shown about the gender and nationality and age difference in different systems. So every country and the population should have their own clinical trials to see the effect and the practice in their own country. So I hope there's going to be many more different, gene therapy clinical trials, not only just for sickle cell disease patients, but many others in Africa and in many other different parts of the world. That's my hope, but I don't know.
I think Daima brings up a very important point. All the developments you are doing are being done in the US, and even preclinical, how do you engage or even have samples from patients outside of the US, especially in low and middle income countries or Sub-Saharan Africa and Asia, where, for example, sickle cell disease is very prevalent, so that you know that what you're developing, at least preclinical, will work in patients in these regions, because this is where the biggest number of patients are. So that you do not just bring those products into clinical trial. Anyway, it's not you, we have to chase you, because we also want these products evaluated in our populations.
Yep.
So I will have, there is a question in the chat. Are there regulatory requirements that must be met before beginning gene therapy treatments? From Brian, a student of molecular biology at Makerere.
Yes. Regulatory system is very complicated and a long process, because you can't just go and say that I have an idea and I need to do this kind of study in human trials. And it's a very long, approval, and there's a very important regulatory systems. And yeah, there is a significant process. And even if you're doing it in the animal setting, like in humanized mice or through non-human primate models, you need to have an approval from the ethical committee. You need to show that this might work and why it might work. So you can't just go and use animals just for an idea. You have to show some positive outcomes that you already got from the cell line studies. And then you can go to the animal studies. Once we have the positive data, then with those data, then you can go and apply for the regulatory agencies to show your positive data, and they'll review it. And it's a very long and painful process, but it's necessary. And the short answer is yes, there is a very strict regulatory system to start a gene therapy study.
Hello. Can I be heard?
Yes.
Thank you for
General
the
Counsel.
presentation. I have two questions. One of it is gene therapy a one type life therapy, or given at multiple times, such that a patient can be cured? Secondly, among these patients who receive gene therapy, has there showed any rejection to the therapy among these patients because of the pharmacogenetic differences? And if so, how have these patients been managed? Thank you.
Yeah. The first question was whether it's a one-time thing or multiple application for the gene therapies. So in most of the case, it's one time if you're doing a transplantation. But as you might remember from the last slides in my presentation, then after transplantation, for example, you're aiming to reach a 20% threshold, but you transplanted a patient, let's say, and a patient remain at 10%, and 10% is not to provide the clinical benefit. So what we developed is try to find a way that you can enrich the edited cells inside the body after transplantation. So in such cases, it can be multiple application, but in most of the cases, and in today's clinical application, it's just one-time thing. So you do the gene therapy and it's done. And generally, if you reach over the therapeutic threshold, the patient is cured. The second question, gene therapy is generally done by the patient's own stem cells. So own stem cells are not causing any rejection, but the problem, a potential problem, is that this CRISPR-Cas is derived from bacteria or archaea. So these are foreign proteins. So if there is a way that they can stay inside the cells even after gene therapy, they can cause some immune responses. But in our non-human primate model and clinical trials with many different kinds using CRISPR-Cas base editing or prime editing nowadays, we haven't come across any immune response to these proteins yet, because we and other laboratories have shown that there is a very limited remaining CRISPR or other foreign proteins inside the cells that do not cause any immune response yet.
Thank you. We have Kenneth online. Kenneth.
Yes, thank you very much for the presentation. It's quite so informative. I want to go back to something scientific, and as well, I think low and middle income countries could benefit from. Picking it from your last slides, when you look at, you talked about ex vivo gene therapy or gene editing, and we saw the prices are quite insane, almost impossible for patients in low and middle income countries there who have sickle cell disease or where it's highly prevalent. And then you talked about the possibility of I think possibility of in vivo gene therapy or editing, which
Right.
might be cheaper and perhaps easily scalable,
Yes.
maybe to populations like the low and middle income countries. Maybe my question now comes from this. You mentioned that we have, I think, some studies which are ongoing, which are trying to use the in vivo gene therapy. Maybe could we hear some of the success stories, and then what could be limiting the same in vivo gene editing or therapy in terms of, for example, in sickle cell disease or maybe HIV, so far which maybe are still making it hard to be picked up. Thank you very much.
Yeah, if I understand correctly, what would be the limiting parameters that to scale up the in vivo gene therapy? But as you summarized, yeah, you're correct. The cost is mostly rely on collecting the stem cells from the body and then doing a manipulation in the laboratory environment. And it's not just a standard laboratory, it has to be very special one. And it has to be, pathogen-free environment. It has to be a GMP environment. And after that, they have to be cryopreserved, and a patient needs to be hospitalized. And after hospitalization, the patient needs to be conditioned. And after conditioning, because the patient is not going to produce new immune cells, the patient is going to be susceptible for infection. So it has to be an intensive care unit for at least 30-40 days. Some patients can stay like six months, and then the transplantation is going to happen, and then the patients is going to be cured. But if there's an in vivo therapy, right, then what's going to happen, it's going to be just one injection. There's no collection of stem cells. There is no, ex vivo manipulation. There's no hospitalization. There's no conditioning. There's no intensive care units. We will skip all these processes. You will go to the pharmacy, for example, and you will ask for an HIV cure. This is a future perspective that how we want it to be. And it's going to be $5 or $10 to be cured for HIV in the long run. And this is possible. And because many studies, as I show in my last slide, that are already doing the gene therapy in vivo for different kind of disease, and we have successful results. So the more we do it, the more we're gonna be expert on this, and we're gonna find better ways to make it available for everybody. That this easy. $3 million is not possible for low-income, middle-income countries, but it's not even possible for developed countries. In the United States, patients cannot pay $3 million for the therapy, so they have to be enrolled in the clinical trials. So it has to go and be cheaper, otherwise it's not going to be possible.
Yeah, we are running out of time. We'll just take one more question from Catherine, but there are many questions in the chat. Selami, what we will do is collect all these so that we can have answers. Again, these will go on the website, both at Capacity and then also the JCRC website. And as we listen to the last question from Catherine, we will, I request everybody who is still on, there is an exit interview after this webinar, please fill it out. I know some people have already left, but we will send emails for this. So over to you, Catherine. You have the last question for this webinar. Can you unmute?
Hello.
Yes, hello.
Muted. Catherine? Unmute. It's supposed to be.
Can you hear me?
Yes.
Okay, I'm saying, what are the future developments and research directions in gene therapy for sickle cell disease? That's my first question. Then the second question is, how long is the recovery period? How long is the recovery period after gene therapy? And what is the long-term follow-up like?
Mm-hmm.
Then the last one.
Yeah, go ahead.
How does gene therapy compare to allogenic HSCT? How does it compare to that? Thank you.
So the future perspective of gene therapy for sickle cell disease is trying to find the best way. Now we have many different options. We have CRISPR-Cas, we have lentiviral transduction, and 2 of them are already FDA approved. And now we have new techniques on the horizon, right? The base editing and prime editing. But consider you're a sickle cell disease patient and you have only one shot. And which one you're going to choose? They are already approved by the FDA and the price are similar. And if you look at the data, they show that both patients resolve, clinical consequences and the severity is gone. They are cured. But you're a patient and probably not in science, so you don't understand exactly what it is, which one you're going to choose. As a patient, there is no way that you can know this, because even the scientists do not know which one is better. So the future perspective should be try to find the best approaches for the diseases, and it should be case by case. So we should explore all the possible therapy options to find the best option for each individuals. And we have now, we can modify the gene in terms of try to reactivate some beneficial protein, and we can fix the mutation. So we have to find what is the best approach. The second question was, how long does it take to get recovered from the clinical trial? So it depends again. It's a case by case, but generally these patients start recovering 3 weeks, as early as 3 weeks post-transplantation, and it takes around 3 to 6 months to fully recover. But some patients can take longer, some patients can take lower. And the third question, I forgot the third question. Can you?
Okay. If you can't remember the third question, again, apologies to everyone. We have run out of time. I think we had very good questions. The questions are in the chat. If your question is not answered, please send it to the email, I think, which was already provided in the link for this webinar, and Dr. Selami will certainly answer all these questions, and then we will post everything so that people can go over even the questions they didn't ask, but to better understand what is happening. I think the bottom line here is what is the way forward. We also have to be involved in the development, the research and development to get products into our populations. And we are not going to sit around and wait for Dr. Selami and others, because I think we have an emergency, we have an urgency, we are the people with the patient. So what are we doing to address the problems that we have in our midst? I would like to again thank everybody who was able to log on and attend this webinar. I would like to thank very much Dr. Selami, who had to wake up very early to give this seminar, and Doctor, we are very grateful, and we will try and get all the questions answered so that we continue to increase awareness, but also educate the scientists whom you can see are very hungry for knowledge on gene therapy.
Thank you very much, everybody.
Thank you.
Thank you. Yeah. Well,